Can somebody confirm you can hear me?
Yes, I can hear.
Thank you. Not sure why that's going like that. All right, my computer's going to be slow because we're still working on the lecture file. It's at 81%. Let me share my screen here if I can find it. There it is. I have three Zoom windows open for some reason. There we go. There, 83% now. Let's get around. All right, any questions before we begin? All right, today is August 5th. Oops. And we're going to be discussing Lab 7, throat and urine cultures. Let me remind you, you have a quiz tomorrow. You can start actually after 11:59 p.m. tonight, so close to midnight. And then you have until 11:59 p.m. tomorrow night to take the quiz. It'll be quiz 3 covering labs 5 and 6 and chapter 4 and the end of chapter 3. Any questions about this? Hopefully I'll send you the unknown project information sometime probably late tonight, probably after 10 p.m. It may even be a little bit after midnight. We'll see how good I get it together and send it out. Any questions about any of that? All right, I think I'm going to close that down because I don't need it. And it'll help speed up things on my computer until this finishes. So lab 7, throat and urine cultures. You do need to read the textbook a little bit before completing this lab. You should read page 392, discussing nose, throat, and mouth normal microbiota. You should also read the beta hemolytic streptococci. It's at the end of this lab module in the appendix. And you should also read about Haemophilus. There's a little bit in the end of this lab module in the appendix. You should also read about the oxidase test. It's on page 132 of your text, and the catalase test on page 155. And then I've added the coagulase test or coagulase production at the end of the lab module in the appendix of this lab. Any questions about any of that? All right, good.
I'm so sorry. I did have a question really fast.
Uh-huh.
Did you mean to finish lab early? We were scheduled to go until 6:50, but you stopped us at 6:20.
Oh, shoot. I stopped you at the regular time. This is not a regular term because this is the summer. I was supposed to go to 6:50. I was wondering when I started the lab, I saw the lecture still there, and we should be in the lecture now. We should not be in the lab, correct?
I do believe so.
Yeah. Let's stop this and go back to the lecture. So, my apologies. You should have mentioned that when I was closing down the lecture, that I was thinking the normal term, and that's we're not in a normal term, we're in the summer term. So I'm going to stop this lab and we'll come back to it at the normal time, which is actually 7. I'm going to start back the lecture and the trouble is I've already got a lecture file.
Why don't you just go to the lecture in this Zoom meeting
I
and
can
go
do
10
that
minutes?
also. The trouble is we'll have a 10-minute break in this Zoom meeting, but I can pause the recording and then have the lab in the lab. I don't know what else to do, so that's what I'm going to do because I don't really want to have 2 lecture meetings today, and I could make another lab meeting, but that would be a little bit difficult. So I think that's what we're going to do. We're just going to continue with the lecture right here. And I'm going to come back and we're right there. So I'm going to put this 2 here, right here. And let's stop that. My apologies. All right, so we discussed this slide, talking about the 4 forms of toxic forms of oxygen. Microbes that use oxygen, and that's all aerobes, produce more ATP and nutrients from nutrients than those that do not use oxygen. So aerobic respiration is the most efficient way to make ATP. Microbes are classified on the basis of the effect oxygen has on their growth. An aerobe is something that must require oxygen, or else the organism cannot grow. And that's an obligate aerobe, meaning must have oxygen or it cannot grow. You can determine the effect of oxygen on microbial growth by inoculating a thioglycolate media with your organism. So an obligate aerobe, well, let me talk about the thioglycolate media. When you have a thioglycolate media and you put it in the autoclave, all of the oxygen will come out of the media, meaning any of the dissolved oxygen will be pushed off by the autoclave. And that's just because when you heat up something, less oxygen will be dissolved in it. And then the thioglycolate media itself will chemically combine with oxygen, removing the oxygen from the media. So when you first take the tube out of the autoclave, there will be no oxygen in the media. But oxygen comes into the media from the air, and it's not shown here, but this cap is actually loose, and so oxygen can come into the tube. That will bring oxygen to the top of the media here, the full oxygen component, meaning 21%, the same as the air. But as you go down the tube, you'll get less and less oxygen because the media will be chemically removing the oxygen from the media. And then at the bottom of the tube, you'll have no oxygen in the bottom of the tube. Any question about any of that? So the thioglycolate media will have an oxygen gradient with full oxygen, meaning 21%, at the very top of the tube. And then as you go down the tube, you'll get less and less oxygen until you get to close to the bottom where there will be no oxygen. An obligate anaerobe is an organism that cannot grow in the presence of oxygen. So you'll only see growth in the bottom of the tube where there is no oxygen. Any questions about any of that? And as you grow up in tube, the bottom of the tube, you have the most oxygen. As you grow up in the tube, you get less growth because there's slightly more oxygen. And as you go up further, you've got one dot there. There will be a little bit of growth around right here because there's not much oxygen, but there's some oxygen, and oxygen will prevent the growth. So you only get a little bit of growth right there. And so as you go up the tube, you have less and less growth. And as you go down to the bottom of the tube, you have the most growth. Any questions about obligate anaerobes and how they'll grow in a thioglycolate media? Now, an aerobe will be the reverse. You'll have the most growth at the top of the tube, and as you go down the tube, you'll get less growth until you get like here, where there'll only be a little bit of growth because there's only a little bit of oxygen. And obligate aerobes cannot grow unless oxygen is present. So you'll have the most growth at the top of the tube and less growth as you go down the tube. In some place about, I don't know, midway, maybe a little less than halfway, you'll have just a little bit of growth, but not much. And then further down, you'll have no growth because there's not enough oxygen to support the growth. And of course, at the bottom of the tube, there'll be no growth because there's no oxygen. Now, a facultative anaerobe is an anaerobe that can engage in aerobic respiration, but it can grow in anaerobic respiration or using fermentation, meaning that this organism can grow in the presence of oxygen, but it doesn't require oxygen to grow because it's a facultative anaerobe. And in a facultative anaerobe, you'll get growth everywhere in the tube, but you'll get the most growth at the top of the tube because it does grow faster in the presence of oxygen, because aerobic respiration is the most efficient in making ATP. And as you go down the tube, you'll get less growth because there will be less oxygen and more of the cells will be growing facultatively, meaning without oxygen. They'll be engaging in either anaerobic respiration or fermentation. And you'll have the least growth in the bottom of the tube, but you will have growth in the bottom of the tube because they can grow without oxygen using either anaerobic respiration or fermentation. So as you go down the tube, you'll get less and less growth, which is hard to see, although there's most growth up here. And then as you go down here, there's not very much growth. And that's because all of this growth is without oxygen. And this growth, all of it, well, almost all of it is with oxygen. And then here we have some with oxygen and some without oxygen. Any questions about any of that? So in the facultative anaerobe, you get growth throughout the tube, but the most growth is at the very top of the tube. And as you go down the tube, you see less and less growth, but you have growth all the way down the tube. We also have aerotolerant anaerobes. These are anaerobes, so they do not need oxygen to grow, but they have all of the enzymes and what is it called? The antioxidants required for handling the toxic forms of oxygen. So they can grow anywhere in the tube because they don't require oxygen, but oxygen doesn't limit their growth because they can remove the toxic forms of oxygen. And there aren't too many organisms that are aerotolerant anaerobes, but of the few they have uniform growth throughout the tube, meaning they'll grow the same at the top of the tube as they'll grow on the bottom of the tube. Any questions about aerotolerant anaerobes? A microaerophile is an aerobe, so it does require oxygen to grow. However, a microaerophile is one that grows best with reduced oxygen. So it grows in low oxygen concentration, but it does not grow in no oxygen concentration, so there's no growth in the bottom of the tube. And then there's less growth as you go from the middle of the tube towards the bottom, and less growth as you grow from the middle of the tube to the top of the tube. So here you get a little bit of growth, and there you get a little bit of growth, but you get the most growth in the middle of the tube. And these organisms usually grow best between something like 10 to 2% oxygen. They do not grow as well at 21% oxygen. There's just too much oxygen, and the aerotolerant, the toxic forms of oxygen start limiting their growth. But they do require oxygen to grow, so they can't grow on the bottom of the tube. An example of a microaerophile is Helicobacter pylori, and it grows in your stomach. There is oxygen in your stomach whenever you swallow. You will bring oxygen into the stomach. But we have other microbes in your stomach which use up the oxygen, and they are either aerobes or facultative anaerobes, which can use oxygen to engage in aerobic respiration. And they'll use up the oxygen in your stomach, reducing the oxygen to around 4% oxygen in the stomach. Helicobacter pylori prefers that oxygen concentration, and that's why it grows well in your stomach. Any question about any of that? So that's how you can determine the effect oxygen has on the growth of various types of bacteria. Let's switch gears a little bit and talk about culture media. In general, a culture media is the word plural for more than one medium. If you only have one, you call it medium. If you have more than one, it's plural, and we use the word media. A media is a liquid or solid solution containing all the nutrients needed for microbial growth. In the lab, we usually sterilize the media and then make it available to you. A sterile media is one that has no living microbes in it. And then you add your inoculum. Those are the microbes introduced into the media that you want to grow in the media. The culture is the resulting media with the inoculum added, where the microbes are growing in or on the culture medium. We have a wide variety of media available to you in microbiology. We actually have a textbook in the lab that's about this thick, about this wide. It's actually a little bit bigger than this book here. This isn't quite a book, bigger than this container here. And, but it shows you that the book with that many pages, on each page of this book, there is one media. And it lists all the ingredients in that media. And it tells you how to make the media as well. So that tells you how many different media we have available in microbiology. So it's a large number. Any questions about any of that? So culture media, we use them to support the growth. And in order to support the growth, you need to provide an energy source in the media, a carbon source, a source of nitrogen, a source of sulfur, a source of phosphorus, a source of trace elements, and if the cells require it, an organic growth factor, like Streptococcus pyogenes. Unless you supply the organic growth factors, you cannot get Streptococcus pyogenes to grow in the media. So these ingredients are needed in a media if the organism requires it. And Streptococcus pyogenes will require all of these, as well as the organic growth factors supplied by red blood cells. The trace elements usually are supplied either in the water you use to make the media, like if you use tap water, or from the ingredients of the media. Like if you have ground up beef extract, the beef extracts will be the ground up cow, and the ground up cow can supply the trace elements. Any question about any of that? Well, here's a question for you. What does a photoautotroph that we're growing in the light that requires, that fixes nitrogen from the air? What do we need in that media in order to get the photoautotroph to grow in the light? Do we need an energy source? It's a yes/no question, people.
Yes.
Yes.
You need an energy source, but this is a phototroph, and then you're growing it in the light, so it does not need an energy source in the
So it
middle
would just need the light.
because it just needs the light, and I'm saying the light is provided. Does it need a carbon source?
Yes.
It does need a carbon source, but it is a photoautotroph, so it can use carbon dioxide from the air. So you do not need to supply carbon in the media, like glucose, a carbon source. So it does not need a carbon source, does not need a nitrogen source, energy source. It's fixing nitrogen from the air. So do you need a source of nitrogen in the media? Nobody wants to take a guess. It's either yes or no. The answer is no, because it can pull nitrogen out of the air. But you would need to supply a source of sulfur and a source of phosphorus in the media. You need to supply the trace elements, but you could just use tap water to get the trace elements.
Now, what if your tap water has like a strong chlorine smell, like a strong chlorinated? Since like where I live, you can really smell that chlorine, and I've had plants kind of suffer from that, and I've had to switch to filtered water.
Okay, what you can do if you have the strong smell of chlorine in it, I don't know where you're getting your water from. It's certainly not
I'm
Vancouver.
downtown Vancouver, actually.
Downtown Vancouver, they have a strong chlorine smell. I didn't know that downtown Vancouver had that. Anyways, what you can do is just have the water sit for 8 hours or overnight, and the chlorine will come out of the water.
Oh. But does it affect any of the trace elements in it?
The trace elements will not leave the water just by sitting.
But they're not damaged by the chlorine in there?
No.
Okay. Oh, thank you.
Okay. The chlorine is just to prevent bacterial growth, and I think most of the bacteria are inhibited. I don't think it's enough chlorine to actually kill the microbes, but I could be wrong. I could be wrong on that. And like I said, if it kills the microbes, that's fine. And then, actually, I had that when I used to live in Houston. Houston is a very warm environment, and the drinking water, they'd pump a whole bunch of chlorine in to keep the microbes from growing in it. And I just didn't like that, so I would refrigerate the water, because it doesn't matter if it's cold water or warm water. You leave it sit long enough, the chlorine gas will leave, and or the chlorine will become a gas and leave the water.
Thank you.
Okay, but you can do that, and chlorine is obviously not a requirement of growth. And obviously the trace elements, well, the trace elements are required, but you can get it in water, tap water, and the organic growth factors would not be required by a photoautotroph, because as an autotroph, it can make all of its needs, usually from CO2 and water, or all the things you need in the trace elements. All right, agar is a complex polysaccharide derived from seaweed, and it's used as a solidifying agent to make a solid culture media. Most solid culture media have agar in it to make it a solid, but you can use another ingredient like gelatin or starch to change the media from a liquid broth into a solid media. But most of them use agar. Agar is usually used in a Petri plate or in a test tube. The tubes, we have two types, a slant and a deep. Oops, make that a little bigger. Oh, nobody said we were going past the time. Let me go like this. So this is a tube showing you a slant. Oops, that's not a good drawing. You get the picture. And then the deep will be a tube like this. And it'll have the agar going across straight. Any question about that? All right, if not, let's end this and I'll give you let's make it a 7-minute break. We'll be back at 7:05 to start the lab. Any questions before I go on?
No.
I'm going to put it right here. All right. I'll be back at 7:05 to start the lab. Can somebody confirm you can hear me?
I can hear you.
Yeah. Oops. I can save that. Let me save that. Get out of here. All right, let's come back to the lab. So we were there. So there are some video clips in today's lab. Upon completing this lab, you should be able to explain how to properly collect throat and urine specimens for routine laboratory culture. You should be able to list the common bacterial pathogens isolated from the throat and urine cultures. You should know the culture media used to cultivate these pathogens. You should recognize which tests are used to identify throat and urine pathogens. And you should understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. We will be briefly talking about the XV factor testing, but you don't need to know it. Any questions about any of that? Oops, too far. Once specimens are received in the lab, they are logged and processed in a timely fashion. Specific culture media are inoculated and incubated to grow human pathogens. Pathogens of throat and human urine specimens. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from the throat and urine cultures. For the throat, we only have two media that we grow our pathogens on: blood agar and chocolate agar. Chocolate agar is only usually used with small children or infants. The main throat pathogen of adults and older children is Streptococcus pyogenes. And this is a throat pathogen of very small children and infants too. But in small children and infants, we also have another pathogen, Haemophilus influenzae. And this bug is not tending to be found in adults. In the urine, we have two media that we always use whenever we grow a urine pathogen. We use blood agar and either MacConkey's agar or EMB agar. EMB standing for eosine methylene blue, And I guess McConkey's could be Mac agar. Blood agar will grow everything, both gram positives and gram negatives. It'll also grow many, not all, but many fastidious organisms. McConkey's agar and EMB will only grow gram negatives. Gram negatives are the main human pathogen found in the urine. And that's why we either use McConkey's or EMB. And then we also use blood agar because the urine pathogens may not be gram negative. Any question about any of that? The main urine pathogens include members of family Streptococcus, family Enterococcus, and family Staphylococcus, as well as species of Enterobacteriaceae. That's a family. The number one pathogen in the urine is actually E. coli in the family Enterobacteriaceae. And then the Pseudomonaceae, the Pseudomonas, is another human pathogen, or they can be human pathogen. Enterobacteriaceae and family Pseudomonaceae are gram negatives, but Streptococcus, Enterococcus, and Staphylococcus are gram positives. Any questions about any of that? All right, for throat cultures, when a child or an adult has a sore throat, and the sore throat has white patches and white on inflamed tonsils, this is indicative of a bacterial infection. The most commonly isolated bacterial pathogen is Streptococcus pyogenes. If you see a red swollen throat with no white patches, it's likely to be viral. If you see the same red swollen throat that has also white patches on it, that's likely to be a bacterial infection. Any questions about any of that? Streptococcus pyogenes is the name I use for the organism, but sometimes it's called Group A Streptococcus or gas for Group A Streptococcus. I don't use these terms because that's an older way. I use the name of the species. Strep throat may be diagnosed by swabbing the back of the throat or their tonsils where there's redness and or white patches. So here and here should be swabbed. You should not swab the sides of the mouth, the tongue, or the roof of the mouth. Patients that test positive for Streptococcus pyogenes are usually placed on antibiotic therapy. And that's because Streptococcus pyogenes has a capsule around the cell, which prevents the host immune system from attacking the bacteria. Meaning the capsule helps protect the bacteria from the host immune system. And that's why often many patients cannot throw off a Streptococcus pyogenes infection that we call strep throat. In the old days, before antibiotics, Streptococcus pyogenes was a main infector of patients, and it caused all sorts of other disease that started with strep throat, but the patient wasn't able to throw it off, and then the bacteria would get inside the blood and would cause red fever, rheumatic fever. It would cause damage to the heart valves and other problems. I don't remember them all because it was before my time. Since Streptococcus pyogenes is fastidious, it will not grow on a general purpose media, such as tryptic soy agar. You need to supplement the tryptic soy agar with 5% sheep's red blood cells to facilitate its growth. We call this plate blood agar, or BAP, for B for blood, A for agar, and then P for plate. And then you streak the throat swab on the plate and you look for any small colonies that are whitish and have a clear zone of inhibition around them, complete zone of inhibition. So this plate does not show Streptococcus pyogenes on it. These are mostly, in this region, are mostly gamma hemolysis or no hemolysis. Let me blow this up. In this region, though, we're seeing darkening of the media, as well as that one there, for example, which you can barely see. Let me blow that up even further. Yeah, that one right there has a dark ring around it, and that would be beta hemolysis, which is very obvious in that region. These, though, that white dot there and that white dot there are actually the colony of bacteria, and then the clear region around it is beta hemolysis of the red blood cells. And this would be Streptococcus pyogenes, a small white colony with the beta hemolysis around it. Now on the blood agar, if you see beta hemolysis, it may not be Streptococcus pyogenes, but there are only 2 further tests that are needed to require confirmation that this is Streptococcus pyogenes. You need to do the catalase test because Streptococcus and all members of the family are catalase negative. And so if it's catalase negative, then it could be Streptococcus pyogenes. And catalase test, you add the bacteria to your slide, you add hydrogen peroxide, and then the cells, if they have catalase in them, they will produce water and oxygen from the hydrogen peroxide, and the oxygen will make bubbling in the on the slide. Most bacteria that grow in water can break down hydrogen peroxide using catalase. However, Streptococcus pyogenes, the members of the Streptococcus family, and one other family, is it Enterococcus? Yeah, Enterococcus, are negative for catalase production. The second test you need, if you have beta hemolysis on a blood agar plate, besides the catalase test, you need to run the bacitracin test. What you do is you take the bacteria grow on your blood agar plate, streak it in this region of the plate, or this region, and you streak it everywhere so it grows on a lawn in this region, and then you put the bacitracin disc, paper disc, down on the plate, and then you incubate the plate. If you see a zone of inhibition around the bacitracin, then this is Streptococcus pyogenes, because Streptococcus pyogenes is sensitive to bacitracin. If there is no zone of inhibition or a very small zone of inhibition, then it is not Streptococcus pyogenes. It's some other member of the Streptococcus family, or maybe an Enterococcus family that is beta hemolytic. And those are the only two tests you need to show that a beta hemolytic bacteria obtained from the throat is Streptococcus pyogenes. Any question about any of that? All right. Another organism that's clinically significant when isolated in the throat is Haemophilus influenzae, which is usually only found in small children or infants. Haemophilus influenzae is a gram-negative coccobacillus. It causes common ear, eye, and sinus infections in babies and young children. Haemophilus influenzae is a fastidious organism that requires the same factors, NAD and hemin, that Streptococcus pyogenes requires. I didn't remember what the X factor was. It's hemin. I knew V factor was NAD, but hemin, okay. However, Haemophilus influenzae cannot lyse the red blood cells, so it will not grow on, boy, what's going on here? Can't seem to move that. Yep, that's not working. Well, I can't seem to move that. Oh well. Let's move it this way so I can just see the picture. Ah, there we go. Oop. Having a hard time moving this. Well, that hit it. Oh well. Anyways, Haemophilus influenzae can grow on chocolate agar, but it normally does not grow on blood agar. The reason why it does not grow on blood agar, which is supplying the V factor and the X factor in the red blood cells, is because Haemophilus influenzae does not lyse red blood cells. I'm not sure what I'm going to do about that. I just want to save this, I guess. Haemophilus influenzae can grow on chocolate agar because chocolate agar is the same as red blood cells. It's just the red blood cells are added directly when you take the media out of the autoclave. So the media are close to 121 degrees Celsius. Is that correct? No. That's the temperature of a autoclave when it's under pressure. But when you take it out of the autoclave, the media will be close to 100 degrees Celsius. And that will lyse the red blood cells. The red blood cells is when the blood agar is when you add the red blood cells to the agar, when the agar is at 55 degrees Celsius or cooler. And that does not lyse the red blood cells. So chocolate agar and blood agar are the same. It's just that the chocolate agar has the red blood cells lysed, and that causes the agar to look brown in color, and that kind of looks like chocolates in it, and that's why they call it chocolate agar. Any questions about any of that? So chocolate agar supplies the growth factors required by Haemophilus influenzae, and so it will grow on the chocolate agar. Blood agar, on the other hand, it will not grow on the blood agar unless you also have Staphylococcus aureus growing on the blood agar. Let's blow this up even further. Here we have Staphylococcus aureus, and you see that kind of off-white streak down the middle of this white area here. That's the Staphylococcus aureus growing on the plate. And then the clear region around that streak, kind of an off-white streak, is the complete hemolysis of red blood cells around the streak of Staphylococcus aureus. If you were to grow the Haemophilus influenzae and streak it on the plate here, going to here, the streak here does not grow Haemophilus influenzae, but in this region, where you're growing the Haemophilus influenzae near the Staphylococcus aureus, the Haemophilus influenzae can grow on the blood agar. The reason why it's growing in this region is the Staphylococcus aureus is lysing the red blood cells here, and then the growth factors coming out of the blood are diffusing up to about here. And that's why Haemophilus influenzae can grow from this region near the Staphylococcus aureus. But beyond that region, in this streak here, Haemophilus influenzae cannot grow. Any question about any of that? Let me shrink that down, go up a little bit. Oh, I didn't show you the catalase test. Here we have hydrogen peroxide, and we put cells in it. The bacteria cells here are catalase negative, meaning they don't have catalase enzyme. And this is a catalase negative test. On this one here, we put the cells in the hydrogen peroxide, and the cells do have catalase, converting the hydrogen peroxide into water and oxygen. The oxygen make the bubbles that we're seeing here, so this is catalase positive. Any questions about the catalase test or the growth of Haemophilus influenzae on chocolate agar? All right. If not, that's it for the throat pathogens isolated from the throat of a patient who has a bacterial infection of the throat. Let's move on to urine cultures. Urine cultures are used to diagnose urinary tract infections and also cystitis, uritis, and polynephritis. Urethritis is inflammation of the urethra, cystitis, inflammation, I believe, of the bladder, and polynephritis, inflammation of the kidneys, I believe. Let me make sure of that one. Yeah, the
Ooh, we
kidney.
can get a deal on that on Amazon.
Sorry?
The first item that came up said you can get a deal with it on Amazon.
Okay, I didn't see that. It wasn't looking. All right, let's move back. So bacteria can travel from the urethra to the bladder, and then from the bladder to the ureters, and possibly up to the kidneys. Although it's more common that the bladder becomes infected than the kidney. females are more predisposed than males to getting a bladder infection. And the reason is just because males have a longer urethra than females. To get a urine culture, the patient needs to clean the genital area prior to collection. And then the patient catches a clean catch midstream involving collecting only the middle portion of the urine stream. You don't want the end of the urine or the beginning of the urine because it'll have more bacteria from the urethra, and bacteria are normally present in the urethra. So everyone has bacteria already in their urethra. If you use the clean catch midstream method, the organisms from the urethra will be less, but they will not be zero. You will get organisms from the urethra, which is just the normal microbiota. A better way to do it is catheterization, but it's a more invasive procedure where you put the catheter up to the bladder. The trouble with the catheterization is it is cleaner, but the catheter goes through the urethra, and the catheter can pick up bacteria from the urethra and then take it up to the bladder. So you may be sampling, once again, bacteria from the urethra. The best way is, of course, suprapubic aspiration, where you put a needle through the abdominal wall into the bladder and then sample the urine in the bladder directly. The trouble is that patients don't like. They're not really fond of the catheter either. And so patients generally would much prefer using the clean catch midstream method. Any question about that? Where did I lose my place? All right. Once the specimen is received in the lab, only 0.001 milliliters, that's 1/1000th of a milliliter, or 1 microliter, that's the same. 1/1000th of a milliliter is 1 microliter, is grown on a blood agar plate. And actually, we have a loop where that right there, let's blow this up a little bit. This loop right there measures out 1 microliter. And then you streak it on the blood agar plate in this fashion, and then you streak this way, getting growth on the blood agar plate. Now, the blood agar plate will grow both gram positives and gram negatives. Most of the urinary tract infections are gram negative, although there are some gram positives. And so we also grow the bacteria on a McConkey's plate or an EMB plate, MAC being McConkey's. And these two media only grow gram negatives. Oops, too far. E. coli is the #1 pathogen isolated from the urine, but there are other coliforms. A coliform is something that grows in your colon, which can be a pathogen in the urine. E. coli is the #1 coliform, though. The interpretation of a positive urine culture is multifaceted. Factors that need to be considered are the type of collection. Was it a catheterized specimen or a mid-catch, a clean mid-catch, midstream urine? Is the gender of the patient male or female? Females actually have more bacteria from their urethra than males. I'm not sure why that's the case, but that is the case. And then the number of colonies per mil of urine and the presence of a polymicrobial growth, meaning if multiple organisms are isolated from the urine culture, it may be indicative of a poor collection technique where there are a lot of bacteria coming from the urethra. All right. Any questions about that? Once we get growth on the blood agar or the McConkey's plate, or both, the gram negatives will grow on both. We then can run the coagulase test. The production of the enzyme coagulase is used to differentiate Staphylococcus aureus from other members of the Staphylococcus family. All other members of the Staphylococcus family are coagulase negative. Coagulase contributes to the pathogenicity of Staphylococcus aureus by converting the blood plasma fibrinogen into fibrin, resulting in a blood clot. The clot actually helps protect the bacteria from phagocytosis, or the immune system, when the bacterial infection is first getting started and the bacteria numbers are small. So a blood clot can help protect the bacteria when the bacteria numbers are small. In the coagulase test, usually they put serum, but you could put fibrinogen with coagulase to make a blood clot, fibrin clot. And there we're looking at a slide test of coagulase. This is coagulase negative, where there is no clumping or clotting. This is coagulase positive in the slide, where there is clumping or clotting on the slide. And here's a serum test in a test tube. If the serum stays, I don't know, semi-solid together in a clot, then it's positive because the clot has formed. If the serum is negative, then the serum will remain a liquid, and when you tilt the slide this way, it'll be a liquid. Any question about any of that? All right, the most commonly isolated bacteria from the urine is E. coli from the family Enterobacteriaceae. There may be other members of the family Enterobacteriaceae which cause the urinary tract infection, and these are gram-negative small rods. There are other gram-negative rods, though, and that's Pseudomonas aeruginosa, can be a human pathogen in the urine. You can differentiate the members of Pseudomonas from the members of Enterobacteriaceae from the fact that the Pseudomonas are all aerobic, non-glucose fermenting bacteria, and they're all oxidase negative. No, they're all oxidase positive cells. Sorry, they're all oxidase positive cells. Whereas the members of the Enterobacteriaceae family are all facultative anaerobes. They're all glucose fermenting, and they're all oxidase negative. The test is usually used to distinguish the members of Enterobacteriaceae from the Pseudomonaceae is the oxidase test, mainly because it's so simple and easy and quick to do. Glucose fermenting, you need at least 18 hours to run that test. Aerobic respiration, you'd need about 24 hours to determine that test. Whereas the oxidase test, if you have fresh cells, can be used immediately. The oxidase test can differentiate bacteria that produce the enzyme cytochrome C oxidase, which Pseudomonas aeruginosa has, because it has the electron transport chain of aerobic respiration. The family Enterobacteriaceae lacks the enzyme cytochrome C oxidase, so it is oxidase negative. What you do is you just put the oxidase reagents directly on the plate containing the cells, or you take the cells on a slide and add the oxidase reagents to the slide. If you see any blue coloring within 30 seconds, then the test is oxidase positive. If there's no blue coloring within 30 seconds, then it's oxidase negative. Any questions about any of that? All right. There's some summary and references. I'm not going to discuss them. Let's talk about the lab. You're not going to be forming any of these activities because everything will be done online. However, you will be held responsible for knowing how to perform these tests and these activities. So know about the Cadlase test. There's a video clip for viewing the Cadlase test. Here's a video clip for the coagulase test. You need to know about the coagulase test. You also need to know about the oxidase test. And there's a video clip showing you the oxidase test. And then answer the questions for the lab module. Let's go ahead and go to the worksheet. So fill in the answers as discussed in Lab 7. Remember, if you get an answer provided by AI, I may mark it wrong. So you're supposed to be getting the information out of Lab 7. For questions 1 to 3, identify the principle used to detect the presence of the enzyme catalase, coagulase, and oxidase. So what enzyme in 1A converts fibrinogen to fibrin? And then name a microbe that tests positive for this enzyme. So you need to name a species name in 1B. For 1A, what enzyme detects the presence of cytochrome C proteins in the electron transport chain? And name 2 microbes that test negative for this enzyme. For 3A, name the enzyme that hydrolyzes hydrogen peroxide into non-toxic products, water and oxygen. And 1B, name 2 microbes that test negative for this enzyme. To say 1B, I meant 3B. 4, which enzyme is used to differentiate between the following? So which enzyme is used to separate Staphylococcus and Streptococcus? Which enzyme is used to differentiate between Staphylococcus aureus from other members of the Staphylococcus family? And then which enzyme is used to separate Enterobacteriaceae, such as E. coli, from the Pseudomonaceae, such as Pseudomonas aeruginosa? For question 5, concerning specimen collection for a routine throat culture, what areas of the throat should be swabbed? And it is 2 or more areas. And then for 5B, what areas of the mouth should be avoided? Also 2 or more areas. 6, what is the most common species causing a bacterial throat infection? So give the species of bacteria causing the throat infection. 7. What test is used by microbiologists to differentiate between beta hemolytic group A Streptococcus, meaning Streptococcus pyogenes, from other beta hemolytic Streptococcus species isolated in a throat culture? And you only need to name one test here. Uh, 8. Answer the following concerning a child's throat culture. State 2 non-selective agar plates that are inoculated to grow the child's throat culture. And what organism can be isolated on CAP but cannot be isolated on BAB? 9. List 2 media that are used to grow urine cultures. 10. List 3 reasons why a urine specimen should be rejected for culture. And you need to see the appendix to get the answer there. Or you could look online. 11. How much urine is placed onto the surface of a blood agar plate when we're cultivating a urine specimen? 12. What is the most common bacteria isolated from patients with a bladder infection? You need to name one species. 13. Identify the Gram reaction, the Gram state, and the cell morphology, the shape, not the growth arrangement of the following bacteria. I think you can find all the information in the lab module, although you may need to look in the appendix. So what's the Gram reaction of Pseudomonas aeruginosa, and what's the shape of the cell? What's the Gram reaction of E. coli, and what's the shape of the cell? What's the Gram reaction of Staphylococcus aureus, and what's the shape of the cell? What's the Gram reaction of Streptococcus pyogenes, and what's the shape of the cell? What's the Gram reaction of Haemophilus influenzae, and what's the shape of the cell? Any questions on anything? All right, it's 7:47. I will be here until 8:30 to help you with the lab. If you have any questions, make sure you ask me before I leave at 8:30 or before the last student logs out. Because when the last student logs out, I'm going to log out as well. So go ahead and start working on the lab. I will be here to help you with any questions.